Hey guys, what's up? Last lab, lab 11. I will tell you what we're going to go over. This is a genetics lab. It's not that long, but it can be a little bit confusing if you've never done it before. We're going to cover a few different components of genetics. Um, the majority of what you need to know is here, obviously. I'm going to go through all of these terms. I'm going to explain to you what certain things are. Um, we are going to understand the makeup of DNA versus RNA. We're going to understand a few processes called transcription and translation, what a gene is, what a chromosome is, and then we're going to practice some Mendelian genetics, aka Punnett squares, of three different types. So the first thing that we're looking at is a double-stranded DNA molecule. It's a double helix, and you can see on here it is made up of nucleotides, and you can see the letters TA, GC, right? So DNA, first off, is two strands. It's made of nucleotides, right? Obviously, the building blocks. A's and T's, C's and G's. We're going to substitute one of those nucleotides out when we talk about RNA, adenine, guanine, thymine, cytosine, and then we're going to sub out one of those for uracil in the presence of RNA. So the main thing that we're looking at here is that they are paired up. Your A's are always paired up with your T's, and your C's are always paired up with your G's. These are called complementary base pairs, and that's just how they fit together, right? Um, when we look at RNA, it will be a single strand, and instead of, pick a different color here, the A's, it's only single strand, but wherever you had a T, you're going to substitute in a U, and respectively the C's and the G's as well. So I will not put that like that. I will put it like that because it's only going to be a single strand. And this becomes very important when we start transcribing um, DNA into RNA. DNA as a whole, right, is a sequence of those nucleotides that we discussed. During during mitosis or cell replication, the DNA will condense into something we call a chromosome. Normally, if you look at a cell, right, the DNA is all just kind of squiggled around in here doing its thing. Things are being transcribed off it. And we call that chromatin. But when we're ready to divide or could force it, the DNA will condense. It makes it easier to move around. But the key factor here is that we have the term gene, hence genetics. And what a gene is a section or sequence of DNA that codes for a protein. Our cells need to make tons of proteins of different types at various times, depending on what they're doing and what their function is. So, how does it know what protein to make? Certain genes would be turned on or activated. So, the most important thing so far is that a gene is a sequence of DNA that codes for a protein. So, the result of all the things we talk about will be a protein. And let's take a look. Obviously, in the nucleus of a cell, that's where the DNA is located, right? Clearly. When you break down the chromosomes, we've talked about this in the reproductive system, we want to have 46 chromosomes. But the key thing here is that, and we'll talk about this when I get more into like the Punnett squares and things like that. We have two chromosome number ones two chromosome number 2s, two chromosome number 3s, and that is because half, right? If you recall, that sperm met with the egg, that's supposed to be a 2. And then they formed a diploid cell, right, which was composed of 46 chromosomes. So one chromosome number 1 came from the mom, one came from the dad, respectively, right? And then our last ones over here will always be the sex chromosomes. Maybe you have a XX, making you female, or an XY, making you a male. These are clearly X's because they look the same. The Y is much smaller, like so. And we call this breakdown of chromosomes a karyotype. So it allows us to kind of see if there's more than one, or I should say, if there's 3 chromosome number 21s, 3 chromosome number 18s, right? We call that a trisomy. Clearly would be abnormal. They'd have 47 chromosomes instead of 46. Or maybe if they're missing a sex chromosome or something like that. So that's the benefits of a karyotype. Key thing, 2 chromosome number 1s, 2 chromosome number 2s, one from the mom, one from the dad. And we'll talk about that a little bit more when we get into the Punnett squares. So, first thing, very important. We have 2 generalized processes of how a gene gets turned into a protein. It happens in a cell. Step number 1 is going to take place in the nucleus. And step number 1 is called transcription. And I have a blank sheet after this. I'll kind of walk you through the basics of it. Transcription is when we take the DNA, whatever sequence codes for a protein, and we more or less copy it into a strand of RNA. We know that RNA is going to be single strand. We know that it's going to be more or less a template of what that gene was. And that process of converting DNA into RNA, it's not converting, right? But transcribing DNA into RNA is step number 1. This RNA leaves the nucleus. Pick a different color for you. And it leaves. And we can see the presence of the RNA here. And this RNA will be turned into a protein. It is turned into a protein by a ribosome hopping on, reading 3 letters, and attaching the appropriate amino acid. And we call this process translation. So 2 steps in converting a gene into a protein, which is extraordinarily important. It's happening in all your cells, most cells, all the time, to make proteins so we can function properly. First, we take the DNA, we transcribe it into an RNA. Then that RNA leaves, goes into the cytoplasm. The RNA is then translated into a protein. Let's take a look, right? We know that our DNA is going to be a double strand. For simplicity, I will draw you 1 strand of DNA randomly, and I will draw the complementary strand. But we're only going to use one of those as the gene. So if this DNA was AAA, TTT, CCC, GGG, we know it's DNA because of the presence of the T, right? The complementary strand, or the other strand, because DNA is double-stranded, would be TTT, AAA, GGG, CCC, respectively. We know A's and T's match, and we know C's and G's match. Now, I'm going to tell you that this is the strand that we're going to transcribe. Cool. So what do we do? In order to turn this, doesn't turn into, but in order to transcribe this, we are going to have to turn it into an RNA, right? That is the process of transcription. So our T will be an A. The A will be a U, and respectively, the C's and G's will be complementary. This is our strand of RNA. How do we know it's RNA? Because there's U's in it. The presence of uracil makes it RNA, right? The presence of thymine makes it DNA. So now we have our single strand. We know RNA is one strand. What happens next? What happens next is the process of translation. Or we will translate this RNA into, if I can write, a protein. What is it going to look like? The first thing we have, I'll do a slightly different color for you. 3 base pairs called codons. And all a 3 base pair means is the 3 letters code for something. And the code that the ribosome is reading correlates to an amino acid. So we know that proteins are a chain of amino acids. So AAA codes for some amino acid. UUU codes for a different amino acid. CCC codes for a different one, and GGG codes for another one. So we have our strand of DNA. We transcribe an RNA. We have our strand of RNA. Each 3 letters codes for an amino acid, and there's a chart that'll show you so you know which amino acid is coded for by which codon. And then the ribosome translates that code into a sequence of amino acids. It folds up, you get your protein. Everything that's supposed to happen in the cell happens in the cell. So you need to be familiar with this, and I'll give you a few examples. This looks like a lot of words, but the reality of it, right, is you have your nucleotides, right, your A, T, C, and G. A process that I did not mention before happens during mitosis or meiosis, and that's called DNA replication. So if I were to go back to here and let's say a cell needed to divide, all you would do was take your strand of DNA and you would just copy it. So if I said replicate the DNA, it would literally look like that. You would just take whatever strand and you would copy it. So DNA replication is DNA to DNA. And we can see that here, right? Complementary strand of DNA. But more importantly, what we were talking about is transcription. That's when we take that DNA, and you could see, as we described, we turn it into a strand of RNA, a complementary strand. We know that RNA has the A, C, G, and U, the presence of uracil. Excellent. And then we have the third part, which is the translation. We can see that this codes for some amino acid, right? And I will show you the chart. This is your practice, but what you would need to do in order to complete this is use the chart. So this is our codon chart. This shows us that UUU codes for phenylalanine. You just abbreviate PHE. You see that AUG will almost always be the start codon, unless we just make up a sequence. Codes for methionine, MET. We see that, AAA codes for lysine. So you would literally just look at the three letters, find them on the chart, and use an abbreviation for the amino acid. So what this would look like, I'll do a little bit of this with you. Step number 1, replicate. What does that mean? DNA to DNA. So the A would turn to a T, the T would turn to an A, the G would turn to a C, etc. Now that you have the DNA, we are going to take the new strand of DNA and we will transcribe it. What does that mean? We take the DNA and we turn it to an RNA. So this would be A, U, G, C, C, G. And if you have to draw lines to practice it so you don't mess it up, cool, makes sense. Now we look at this 3 base pair sequence is going to code for an amino acid, right? So we look on the chart, AUG, we know stands for or codes for methionine. So over here, we can put it at the bottom. You can write it inside. It doesn't matter. CCG. See if we can find CCG. Codes for proline. So we would write pro. So we have a couple basic concepts. The nucleotides that are involved in a double-stranded DNA versus a single-stranded RNA. We have the topic of replication, which is copying a DNA to a DNA. We have a process of transcription, which is clearly turning a DNA into an RNA. And we have the process of translation, which is taking that RNA sequence and organizing the correct amino acids in the correct order. You will eventually get to something that says stop codon. And that would be, let's see if we can find a stop codon. Boom. UAA, UAG, UGA. That ribosome would hop off and basically the protein's done. Right? So this one here is talking about a mutation. I'll discuss it, but you don't have to stress too much about it. Things can happen. In this case, a deletion mutation. Right? So this would be in position 8 of the new DNA strand, and we know that because it says it right there. So all you would do, if this was your C, your A would be a T, this would be a C, G, T, A. When you transcribe that, pretend that that position 8 is not there. It's like if you're typing on a keyboard and you just deleted something, everything would slide down. So your T, right? So that'd be G. This would be A. It would just slide down. So this would be a C, right? Because the G would turn to a C and then everything else would be the same, right? So you'd have that. Obviously, I'm doing it on the same line, which is probably not wise, but that's all good. So that would slide down. Then obviously, if that was that, the T would turn to an A, and the A would turn to a U, respectively. So all you did was just delete that one base pair. We call that a deletion mutation, but I'm going to get rid of that just in case you want to look at this sheet complete, to kind of get an idea how to do transcription and translation. I give you some examples here, right, to practice. And that's the first of 4 major components in this lab. Got to understand that, know your nucleotides, know the composition of DNA, RNA, first proteins. I do not make you memorize that chart. Anytime I ask you a question, I will give you the chart or ask you something oriented around it. You do not have to memorize all those codons. That would be crazy. There's 64 of them. The last topic / is broken into 3 components, are Punnett square problems. So we have three general types of Punnett squares I'm going to describe to you. The first one is what we call autosomal, or just regular dominant recessive type Punnett squares. The second one is a sex-linked, or in our case, we will discuss an X-linked Punnett square. And the third one is a codominant or blood typing problem. I will give you an example of each. And then obviously I have a bunch of practice problems for you to go through and kind of practice that. I'll probably ask you some questions. First one, autosomal. And I'll walk you through kind of how I go about doing this, my thought process, and possibly how you should also do it. So I always start off like this. We will have two alleles. So the first thing is that we have dominant alleles and we have recessive alleles. If we recall back, I said you have two versions of chromosome 1, two versions of chromosome 2, two versions of chromosome 3. So wherever that gene is located, you're going to have a version from the mother and a version from the father. And obviously, if you're looking at the mom, she has parents, so maybe she got the dominant allele, which means we will express the protein from her mother, and maybe she got a recessive allele, recessive are always lowercase, from her father. And maybe with the dad, he got a dominant allele from his mother and a recessive allele from his father. It doesn't matter. We'll always tell you kind of what they are. This makeup where you have a big A, little a, is called heterozygous. Other scenarios would be big A, and we call that homozygous dominant. And little a, which is called homozygous recessive. So these are our three allelic makeups. Big A, homozygous dominant. Big A, as I described here, heterozygous, right? Homo means same dominant. Hetero means opposite, big and a little. And homozygous recessive means little a. So perhaps I say the mother and father were both heterozygous. This is what we would start off with, right? So tips. Always write out what the homozygous, or I should say, what the dominant and recessive allele mean. So I'm kind of just giving you this all at once, but then we'll do an actual problem, right? So we know big A is dominant, little a is recessive. Let's say in this problem, the dominant gene coded for, you could just make something up. I don't know, tall. That's polygenic, but it doesn't matter. We're just making something up. So big A would mean tall, and it would say it somewhere in the problem. Little a would be short. It's not how it actually works, but we're just practicing. So the mother would be tall. The dominant allele always makes a protein, thus she's making a protein and she will be tall. The father is also tall. However, both of them are carrying a recessive allele. So when we do our Punnett square, and it doesn't matter who goes where, as far as, mother, father, top side, doesn't matter. We're going to look at if they had offspring, half of the sperm would have a big A, half of the sperm would have a little a, half of the eggs, in theory, or one of them, would have a big A or a little a. So the possible genetic makeup of their offspring would be this. So a couple keywords. One, I always write out the mother and the father, 2 slots. Let's fill it in. Maybe after this, I'll do another example. Two, I always want to figure out what the dominant allele codes for. An allele is just a different version of the same gene. So you have a dominant version and a recessive version. The big A codes for tall, the little a codes for short. So now we have to give the answer. The answer is, what is the percent homozygous dominant, percent heterozygous, and percent homozygous recessive? This is the answer. So we're going to say 25%, right? This is what we call our genotype. And then I will show you phenotype. I kind of ran out of room, but I'll draw it somewhere. 25% chance their offspring would have a big A, and thus would be tall. 50% chance, do it in a different color so it's not confusing. Heterozygous. It would still be tall, right? And 25% chance homozygous recessive. This is what we call the genotypes, the percentages of what the allelic variation is going to be. The other part is called a phenotype. And the phenotype is what are the percentages for the physical traits. So here, what are the percentages that the child will be tall? And what are the percentages that the child will be short? So we can see here that presence of a big A means they're going to be tall. Tall, tall, tall. So we have 75% chance tall and 25% chance that they are short. Big picture. Genotype, the letters. Phenotype is the physical expression, or are they tall? Do they have the disease? Are they deaf? Whatever. What type of blood do they have, etc. So this is what we call an autosomal, just standard, dominant, and recessive. I don't want to erase this because I want you to be able to look at it. We have a couple keywords: heterozygous, homozygous, dominant, homozygous recessive. And the way I go about doing this is always the same, right? Step number 1. This. I draw the 2 slots for the mother and the father. Step number 2, I figure out what is the dominant and recessive gene coding for. Step number 3, I plug them in. Step number 4, I do the Punnett square. That's kind of how I go about it. One step I often see people miss is this. This is a very important step because you have to know what the big letter and the small letter are coding for. So you should be familiar with these, be able to do them. I can ask you, what are the genotypes? How many of their offspring would be tall? If they had 4 kids, 3 of the 4 would be tall, in theory. 1 of the 4 would be short, and I can ask you questions like that. That's first type of Punnett square. The next type of Punnett square is an X-linked. So the difference here, what do I do first? I draw my mom and my dad. The difference with the sex-linked, specifically, this is an X-linked problem, is we start off, that's not the mom. Before we write any letters on top, we start off with their sex chromosomes. A female is always XX. A male will always be XY. The most important thing about this is that this is an X-linked condition. Important. So the letters, let's say big A, little a, are only on top of the X because the disease is linked to the X chromosome. So there will be nothing up there on the Y. Don't write anything on the Y. I don't show you Y-linked diseases. We're focusing on X. So let's say here, I'll just do the same thing again because who cares? Tall or what? Let's do disease. Right? Normal disease. In this case, the dominant gene makes you normal. The recessive gene gives you some disease. Let's say that the mother was normal and the father was normal. That's what we would know. But let's say the mother's mom had the disease. Could be that. So you see we have that little superscript, that letter above the X. Nothing above the Y. So let's do our Punnett square. These two have children. First kid comes out like that. No disease. Second kid comes out like that. No disease, but they're a carrier. Both of those are females. Next kid comes out like this. Next kid comes out like that. So in this situation, and very commonly, you will see an X-linked condition more likely found in a male because they only have one X chromosome. So if that one's messed up, they have the disease. So what are we looking at? We could do our genotypic frequencies or our genotypic ratios, right? Genotype, it would be 25% all. That's a Y. 25%, 25%, 25%. But what is our phenotypic ratios? That would be normal, right? 75%. Disease, 25%. So the main difference between the first one, which we did, was an autosomal, just normal, recessive, and dominant characteristics. With this one, you have sex chromosomes. The disease is located on the X. Nothing's on the Y. The male only has one X, so he's more likely, if that gene gets passed down, to express it. He doesn't have another one to cover it up. So in this situation, we would have one male, obviously, that has the disease, 25% chance. The other females would be normal, although one of them would be a carrier for said disease. That is the second type. So we have transcription translation problems. We have the normal autosomal or regular practice that we just did. And then we have the second type, aka third question, which is the X-linked or sex-linked Punnett square. And the last one, type of Punnett square number 3, deals with blood typing. Talked about blood typing way back when, 8B maybe. We're not going to go into as much detail with it, but we're going to talk about the genetic component of it. So your options, right, are A, B, or nothing. We'll call it O for now. Sometimes they use an I, same difference. So the A and the B are dominant alleles. The O is the recessive, which means we don't make a protein, which we discussed way back when. So if the mother is blood type O, she must have two O's. If the mother was blood type A, it could be AA or it could be AO. So we would have to have some more information. Let's say her mother was O. She would have to be AO. We have to have another piece of information to understand what her second allele is. If the father was blood type AB, it would only be AB. If he was type O, it would only be OO. If he was type B, we'd have one B and we'd have to figure out what the other letter is. Hopefully that makes a little bit of sense. So in this scenario, let's break it down. AO, AB. What are the genotypic frequencies? Right? 25% AA, 25% AO, 25% AB, 25% BO. What are the phenotypic frequencies? These are blood type. So this would be type A, 50%. Why? Both of those are type A. There's no RH, there's no negative, positive in these problems. We just do A, B, and O. AA is blood type A, and AO is blood type A. Because if you have an A, you make the A protein, your blood type is A. 25% chance AB, and type B, 25% chance. So let's say these two parents had a kid. Could that child be type O? No. If that child was type O, that's not the dad. Could the mother have a child that's type O? Yes. So maybe, I don't know. That guy's not the dad. And that's kind of the practice that you would go about doing it. So this is called codominance because you can express two dominant alleles. You can have an A and a B, both making proteins, both being dominant. Kind of makes sense for all the problems. Establish the two slots. Establish what your dominant and recessive are. These are your dominants. This is your recessive. Plug in your Punnett square. First, find the genotypes, obviously, of the parents. Plug in your Punnett square. Be familiar with the genotypic and phenotypic percentages. That's your third type of problem. Everything else you have on here, right? And I gave this lovely sheet. You could print it out. You could practice it on here. You could write if you got a touchscreen. Everything else on here is practice questions. The hard thing about it is their wording. So you have to be able to read through each one of these questions, figure out what it's asking, right? You could see here, this one is a regular autosomal, right? I'll do this with you. In a particular breed of dogs, deafness is an autosomal recessive trait. autosomal recessive, deafness. So I put the big D, normal, little d, deaf. Pretty straightforward. Now, two dogs are mated. Cool. One dog is deaf. What does that mean? One dog has little d, right? The other dog is normal, but one of its parents was deaf. That tells me that that second dog has to be big D, little d. What is the probable percentage of normal and deaf dogs that might be found in the litter? Give the probable distributions of genotypes and phenotypes. Cool, we got that. So as long as you set it up right, you'll probably be okay. Big D, little d, big D, little d, little d, little d, little d, little d. And this gives us genotypic, right? Big D, little d, 50%. Little d, 50%. Phenotypic, we would have 50% normal, 50% deaf. And you go through and you practice, right? This is a autosomal dominant trait. That means that this type of cancer, whatever, the big R, if you're using an R, the big R is the disease. Autosomal dominant, the little r is normal, right? A heterozygous woman, big R, little r, was successfully treated for this disease. She has a husband that's normal, little r, and then you just do the Punnett square and describe it, right? Huntington's disease, autosomal dominant. Same thing, right? We just talked about PKU, autosomal recessive. This is telling you that these are all autosomal problems, aka the regular one, and the disease for PKU is on the little letter. The big letter is normal. Two phenotypically normal parents have one kid with the disease, right? Autosomal recessive. Phenotypically normal means that they have to have a big letter, but they have a kid with the disease. There's probably bad letters to choose. Let's do A. So you'd have a double heterozygous cross. Two phenotypically normal parents. They both have to have a big letter. Had two children. One was normal. Cool. One had the condition. Thus, they both must have a small letter. We do the Punnett square. Hemophilia, X-linked. That tells you that this is an X-linked problem. So the first thing you do, you draw your XX and your XY respectively. And then hemophilia H, right? X-linked recessive. A woman has one hemophilic son. So she's got the little h. We're assuming she's normal, right? And her husband's normal. So you would do that, Punnett square it out, do your genotypes and phenotypes. The last two are blood typing questions. Number 8 has multiple parts to it, but you get the gist, right? I'll help you with this last one. In a paternity suit, blood types are found to be mother A. That's all we know. Child O says it's got to be that, right? Because she had a kid that's O. Obviously, she has to give the O, so she's AO. Alleged father, AB, right? Are there adequate grounds? Who's who, right? Is this his kid? You do it, you get an A-O, you get an A-B. Don't forget the child is O. Is there any chance that this is an O? No. You get your answer, right? Hopefully that makes sense. The three main topics, as I described, two main topics are transcription, translation, and understanding genetics. I'm going to ask you some questions on that, as well as Punnett squares. And obviously, I'll have to ask you probably multiple choice questions about it. You won't have to write them out. The three different types of Punnett squares, um, understand genotypic versus phenotypic percentages, what they mean, and the differences between the three different types of Punnett squares. Um, Other than that, you have your last test. Kudos to you. That's the last lab. You will not see me for another lab, probably just a couple lectures.